it's a real great pleasure to come and give a talk today. What I'm going to talk about today is about the new emerging observations of how dramatic genetic mosaics we are, and really to the fundamental question of what is it that drives mosaicism in our tissues. So I always begin with this slide. Of course, you all know these two figures, 71 years ago, but you probably have never seen this photograph because actually it shows CRUK repairing DNA here. And he isn't doing it for any other reason other than to take the famous iconic photograph. But what I want you to recognize is repairing DNA damage is pretty fundamental to life. Now, here is the iconic molecule, and what you may not all be aware is that in all our cells, as the DNA is sitting in the nucleus, it's constantly being attacked and changed. And it's the cycle of these changes that engages DNA repair. The base damage that you have is constantly being fixed and reversed, so you retain the coding information of the hard disk of the cell. But every now and then, misrepair happens. And this misrepair manifests itself in point mutations, okay? And what I'm showing you here is the very infamous mutagraphs that were invented at the Sanger Center. And this is a mutograph that's present in all cells in our body, and it's got some very unusual properties which I'll go on to tell you about. But just to orientate yourself, these are the base changes that occur in a single cell in the genome. The height of each of the bars represents how often that particular changes, and the fact that there are many bars for any of these changes is each bar represents the 5' nucleotide and the 3' nucleotide from the one that's altered, okay? So that's what a mutograph is. Now, I like to use the word the mutation seismograph because it's a kind of indication of the way the physiology of the cell alters DNA base changes. And as I mentioned to you, this is probably the most famous of them and least understood of mutation signatures, which is mutation signature 5. So just to orientate you on what this mutation signature 5 is, it's present in every single cell in the body. So whether you're a nerve cell or a dividing cell, it'll have this mutation signature inside it, right? So you don't need to copy DNA in order to get this. And when this was first put out in 1920, about a couple of years ago, it was quite an earthquake in the DNA repair field because it was always thought that in order to put a mutation, you needed to copy DNA. But this clearly happens in the absence of copying DNA. There's another remarkable property of mutation signature 5. So if you take the same cell and you look at it, which is the gut enterocyte in this instance, in a mouse, a dog or a human, the rate at which mutation signature 5 happens is a function of time, and it scales to the life form. So mice do it much quicker than humans, okay? And so in a way, it's a very good chronograph for lifespan. Now, we don't know, and the really big question is, what is it that causes mutation signature 5? And I'll come to that because that's the main purpose of my talk. But one key element of it is the price of metabolism, the fact that the cell respires and so forth and does various metabolic transactions that ultimately attacks DNA and introduces these mutations. But it is a very big open question. But I hope I'll convince you at the end of my talk that this is a tractable question and an important one to address. Now, each cell in our body has mutation signature 5 operating in it, and those mutations are throughout the genome. And an important realization of this was that each cell therefore has a mutation barcode in it. And this concept of the barcode was very cleverly exploited by my colleagues at the Sanger. And what they were able to do by taking a tissue, a small tissue from any part of your body, and sequencing it to depth, using the barcodes of each cell, they were able to lineage trace how a single cell evolves over a tissue plane. And I'm going to show you two examples that are ostensibly related to as we age that demonstrates the power of this approach. So the first example is the human esophagus. And so in this paper in Science, the Sanger groups, what they did was they took a small patch of the esophagus and they ultra-deep sequenced all the DNA in that small patch. So there are maybe about 10,000 cells in that patch, or it's probably about 100,000 cells. And what they then did was they computed onto that, with the frequency of those mutations, those barcodes, a virtual map of how each individual cell competes with its surrounding cells. And what you can see here is such a virtual map of an esophagus in a young individual. And what the white represents half mother, half father, so the majority of cells haven't diverged, but the bubbles represent a single cell which has now started to compete in that space. Okay? Now, if you do this experiment in a young person, and you compare it what happens to when you get old, by the time you're 70 years old, this is very striking. Your entire esophagus is a tapestry of competing and warring clones. And it's really striking that actually so much has diverged away from mother and father here. And this is mosaicism, somatic mosaicism driven by mutation, occurring at scale and altering a tissue in quite a fundamental way. In this is a deep paradox in cancer research, because many of these big clones that have started from an original cell, ostensibly normal esophageal cells, have all the genetic mutations that theoretically would make them cancerous, yet they're not cancerous. And that is actually a big fundamental question in cancer biology, which is what wakes up all those silent cancer genomes that exist throughout our body and all our tissues. In another example, which is a bit more relevant to my work, they did the same sort of analysis, this time not in a tissue, but in a liquid system, which is blood. Essentially the same principle. You're using mutation signature 5 barcodes in individual cells to then compute the lineage of that tissue. Okay, so and this is represented here. So when you do this sort of analysis in a young individual, what you get are many all the stem cells over here giving rise to daughter cells. So you get a very nice tooth comb of lineage, right? However, if you do the same sort of analysis in an old individual, what you see is a dramatic degradation of this. And what this tells you is that whereas many stem cells contribute to the production of blood in a young individual, by the time you're 81 years old, almost half of your blood is made from one ancestor, one stem cell, which has acquired mutations that has given it a selective advantage. And this phenomenon is known as clonal hematopoiesis. It's present in about 20 to 30% of people over the age of 70. And we now know it's a very strong predictor of getting blood cancer. It's a strong predictor of failure of blood production as we get old. And it's actually a strong predictor of vascular disease, which is not fully understood yet. So, this is tissue mosaicism as a consequence of mutation operating in scale over time as humans age. Now, the principle of mosaicism has now been extended to many tissues. As you'd expect, it operates very strongly in skin, and that's not a surprise because a lot of the damage or the mutation induction is coming from the outside with UV light. But it also happens in the liver, and in a beautiful paper in Nature, the Sanger group showed that if you have fatty liver or alcoholic liver disease, you select hepatocytes to ward off the influence of sugar, fat, and alcohol. So you actually develop clonal hepatogenesis in your liver. And I think that's an important phenomenon because most people in endocrinology would not think of what that means to the physiology of something like handling sugar in diabetes or type 2 diabetes and so forth. And I think the fact that we have such dramatic genetic mosaics should make us rethink how we think of human physiology over time. In blood, mosaicism is very prevalent. Almost 84% of the mutations that give rise to clonal hematopoiesis have arisen from mutation signature 5. Okay? So a really big question in the field is how can we understand what drives mutation signature 5 and if possible, subvert it. Okay, so You'd have to go back to 1993 to a very famous review written by Thomas Lindell, who's the founder of this field. Thomas went on to win the Nobel Prize for discovering this field. And what Thomas postulated was that DNA is intrinsically unstable because it lives in a bath of water and oxygen. So hydrolytic damage to DNA is probably one of the main drivers of the base adduction that ultimately leads to mutation signature 5, right? But I hope by the end of my talk I'll convince you of 2 new molecules you should put in this category. The first is formaldehyde, which you know as a chemical that's used in biology quite a lot. And the second you'll probably be acquainted later on this evening, which is acetaldehyde, which is the byproduct of your gin and tonic. And we've published extensively to show the role of these 2 factors. So how did we come to realize that this matters? It really comes through the lens of 2 very dramatic human genetic illnesses. One of them is called Fanconi's anemia, and these young individuals get blood failure because they get clonal hematopoiesis very early on in life. That eventually leads them to losing blood production totally or dying of leukemia. But if they survive that, they get multiple cancers of other organs that normally people in their 50s and 60s get, except they get it at the age of 20s. Okay? And then there's another human genetic syndrome called Cockayne syndrome, which is quite the reverse of this, in that in this instance, people prematurely age. So they get neurodegeneration by the time they're in their teens, they develop kidney failure and a very striking cachexia. You see this in this 24-year-old person with Cockayne syndrome. And in both these diseases, they're DNA repair defects. And the main thing that they cannot fix is a DNA crosslink, something that a form of DNA base adduction that glues the 2 strands of DNA together. Okay? Now, from our work, what we've been able to show is that the main reason, the main agent that crosslinks our DNA is actually this compound formaldehyde. And the reason you get the difference in the disease pattern is that the children who get the bone marrow failure and Fanconi's anemia, if you have formaldehyde gluing the 2 strands of DNA, you can't separate that when you copy DNA. So you need to have a specialized repair system that removes that, okay? So that's why the defect manifests in fast-growing cells, like the bone marrow. However, in Cockayne syndrome, when you have a DNA crosslink caused by formaldehyde, you don't really care about it unless it interferes with transcription. You need to read the DNA. And in fact, you need to remove that by the system that goes wrong here in order to be able to transcribe DNA. And that is why tissues that don't divide, like the brain and some aspects of the kidney, are the ones that degenerate. So I think that explains potentially the physiology of how these diseases arise. So turning to formaldehyde, I need to tell you a little bit of where it comes from, right? Well, we don't really know the question of where it comes from, but what we know is that every organism in all kingdoms of life, all the way from bacteria to humans, have evolved a system to remove the formaldehyde before it even gets a chance to access DNA. And there are 2 key enzymes that do this, okay? So even before you need to engage DNA repair, you have 2 enzymatic systems that clear it. And that's essential in life. So a couple of years ago, what we did was remove these 2 enzymes really and ask, if you took this away in an animal or if there were human examples, what would happen? Could the 2 DNA repair systems that operate to fix the crosslinks that they create, would that be enough to get you out of that? And in fact, it isn't, because in fact there are children that are born in Japan, and the reason of Japan in East Asia is because they already carry 500 million people in that part of the world carry a mutation in the first gene, which is the basis of the Asian flushing mutation. But then if they lose a second gene, they lose their ability to clear formaldehyde. And what happens is that they develop a phenotype, which is the intersection of both diseases. They get the bone marrow failure and the premature aging. So we have perfect genetic convergence, which is we have a substance that our body is producing. If you can't remove it, you need these two repair systems, okay? And there's a metabolic route to the same phenotype as the DNA repair deficiency. So it does fit pretty well. Now, obviously, the humans are rare, so we really originally came to this discovery by actually deleting the two enzymes of clear formaldehyde in mice. And what you can immediately see is that it's essential, right? And the few animals that survive get a very striking premature aging phenotype in that they don't live very long, they become very wasted, lose muscle mass, and eventually die of bone marrow dysfunction or cancers in multiple tissues. Now, how do we know it's formaldehyde? This experiment took the better part of 5 years to do, which is for the first time to reliably measure the blood level of formaldehyde. And we collaborated with atmospheric chemists in the University of Leicester to do this, using gas phase chromatography. But what's striking is that in mice, wild-type mice, the blood level of formaldehyde is about 2 to 3 micromolar. That's really high when you think about that, right? So there's a significant amount of formaldehyde produced. But if you lack the protection system, it's about 50 to 60. So about 100 micromolar of formaldehyde is embalming levels. So I find it pretty remarkable that there are humans that can walk and mice that can crawl along in life with sub-embalming levels of formaldehyde in their blood. But what this really tells you is that actually the body produces a lot of this formaldehyde and that you need to clear it. Now, I began my talk about mutation signature 5. So how do we know that formaldehyde adducts DNA? And we can do this by actually looking at a biomarker of formaldehyde exposure in DNA and the modification of one of the bases, guanine. And if you do this, you can see, using a log scale here, that the amount of DNA base adduction that happens in mice and humans that can't clear the formaldehyde is about 10 to 15 to 20-fold higher than compared to the respective controls. So whatever is the excess formaldehyde that's produced in the body, it gets access to DNA. Well, can we provide any evidence that it has anything to do with the famous mutation signature 5? So in order to do this, by the way, this is actually a very expensive experiment to do because you have to take the bone marrow stem cells and grow each colony, and you have to sequence about 1,000 of these colonies and extract a mutation signature to get statistical power. So if you do this, thankfully, in the wild type, in the normal situation, you recover mutation signature 5. Now, what happens when you have formaldehyde excess in the system? And this mutation signature that you extract. So what will immediately be apparent to you is that, like 5, the amplitude of all the mutations, point mutation bars, goes up. It isn't a perfect match to mutation signature 5 because there are some elements that are very distinct, but it's a very close approximation. And I think this is an important experiment, at least for my lab, because it for the first time shows that really figuring out what drives this fundamental mutation signature, which is a barometer of lifespan to some extent, is tractable, and you can get to it and understand what constitutes it. So I hope I've convinced you that other than water and oxygen, you need to think of formaldehyde in that category, and there are probably many other base modification agents that ultimately drive mutation signature 5 and contribute towards aging and mosaicism. So the real question is, where does formaldehyde come from? So I hope I've given you good evidence that it adducts DNA and that there's an addiction to two repair systems in order to fix the damage it comes from. Now, it turns out that there's a very fundamental biochemical reaction, which is oxidative demethylation. Oxford's a very famous place for this type of biochemistry because that's actually the basis of many of the enzymes that work with oxygen sensing. So whenever you modify histones or DNA, you spin out formaldehyde very close to DNA. And so that immediately explains why you need to have these very powerful systems to restrain their activity. But is that all there is to it? Now, I'm summarizing a lot of work from my lab. We know that demethylation, as I described, is a contributor to formaldehyde. In a paper in Nature we published in 2018, we showed that decomposition of the the vitamin folic acid actually releases formaldehyde. And we were able to show that a source of formaldehyde comes from cells from the activity of mitochondrial complex 1 through genetic squeeze. But none of this can really explain the 2 to 5 micromoles that's in blood, because these are operating probably at the nanomolar or much lower concentrations. So there must be a significant source of formaldehyde coming from somewhere. Now, I think a clue, and this is work that's still ongoing in my lab, a clue could be the phenotype that these people manifest. So the children that accumulate excess formaldehyde and individuals with Cockayne syndrome spontaneously develop severe cachexia. And in the case of Cockayne syndrome, and also these children, many of the children don't eat at all, so they have to be tube fed. So maybe this is a physiological circuit that's putting you off food. In a way, this is a form of genetic anorexia nervosa, right? And that's what we are trying to chase. And just to show you how dramatic this is, in a formaldehyde superaccumulating mice, they're almost half the size of normal mice. And if you look at the adiposity, there's hardly any fat in them, right? So we don't know the full answer to this, but what we do know is, at least in experimental mice, the effect of formaldehyde is greater in males than in females, and there's sexual dimorphism with the rate we age and the rate we have mutations, so that somewhat fits to some extent. If we take our mice and we restrict them by 30% caloric restriction, which is a very powerful way to extend lifespan, the level of formaldehyde adduction in DNA reduces, the biomarker we've developed. And if you get rid of the gut microbiome, that also happens. Okay, so there is something from food or an exogenous source interacting with the bacteria in our body that contribute to the production of the vast bulk of formaldehyde in our body that's present in us. And we have some preliminary evidence that if you feed the mice a lot of pectin, because there's an enzyme that bacteria have that convert it to methanol, which is then broken down into formaldehyde. But I wouldn't want to give out a view that eating fruit is bad for you. So I'm now looking into the future. I'm pretty convinced that formaldehyde is a driver of mutation signature 5. Mutation signature 5 is an important thing to understand if you want to understand how DNA, aged DNA comes about. But a really important question is, how do you get to the other agents that damage DNA and cause base adduction? How can we detect adults? And some of the things we're doing here is basically we're adapting mass spectrometry. So this field of mass spectrometry has transformed our ability to, in an unbiased way, to identify proteins, for example. But the ability to do the same, use the same technology to look at DNA in an unbiased way has actually fallen away, because actually very few people work on mass spectrometry of DNA. And so we're developing techniques that might allow us to do this, right? And we know this works because that's how we identified the formaldehyde adduct in the first place. So, that's one of the things we're trying to develop. The other way, theoretically, it should be possible to use nanopore sequencing, because what nanopore sequencing requires, invented in Oxford, in fact, is it uses the signal that's given off as a base traverses into a channel. And if you have an altered base, it should, in principle, give you a different trace. So we hope to be working with Oxford Nanopore to try and develop ways to do this. And what I think we're really lacking in this field is an ability to look at DNA health in us at single cell resolution, but also at scale. Because there's no question that this is an important thing to do, but it's kind of been forgotten to look at. So just to summarize, I think I'm reiterating that you could put formaldehyde in this list. And then finally, just to show that it must be important, there must be a lot of it produced in us. Some of it does come from the outside, because evolution has devoted a lot of effort to try and restrain its ability to attack DNA. There are 2 enzymes that clear it, and there are 2 repair systems. And there are a remarkable number of human syndromes that contribute to this. And I put this down here because this actually is one of the most common traits in man, which is the mutation of this enzyme, which we know clears formaldehyde. and also does the aldehyde that comes from the 2-carbon version, which is acetaldehyde that comes from alcohol, which is the basis of the flushing mutation in humans. But I think its main purpose is to take care of formaldehyde. So that's about 500 million people who are partially deficient in this first tier. And in fact, we know that those individuals are completely reliant on these two repair systems, because if you take them away in humans, then you get a very accelerated phenotype. So finally, I'd like to acknowledge the people who've done this work. I have a great team here in Oxford who've done the majority of this work that I've described here. And of course, I've had some very good support from Cancer Research UK, the Wellcome Trust and the MRC to push this work forward. Thank you very much for your time.
Absolute tour de force, as ever. Thank you very much. Now, questions from the audience. Joe, there's probably a mitochondrial DNA question coming up.
Yes, there certainly is. I'm sorry, I probably had a mini sleep when you introduced what mutation signature 5 meant. And of course, my supplementary is, and does it involve mitochondrial DNA?
So you don't see signature 5 in mitochondrial DNA, and it's unclear the reason why. But also calling mutation signatures in mitochondria is much more difficult than in the nuclear genome because of the heteroplasmy effect. So we don't know the answer to that question. So you've got a follow-up question.
But you didn't say exactly what mutation signature 5 was.
So mutation signature 5 is a mutation pattern which has a spread of all changes in all 4 bases to the opposite, either purine or pyrimidine. It occurs in a very precise pattern, and it's present in every cell in your body, and it increases, the amplitude of it increases as time goes on. And animals that are short-lived do it much faster than animals that are long-lived.
So it's basically just the base changes.
It's just the point mutation base.
Human clone or anything like that.
Sorry?
It's not one specific family type or
No. It's, in a way, there are many processes, many agents that are driving it, and there are many repair systems that are contributing to it. But the question is, can you deconvolute it? Can you identify what is driving it? And
but so when you said it's 84% of things, 84% of something arise on the mutation signature 5 background, I kind of thought that must mean that some of the population didn't have mutation signature.
No. I may have confused you. If you now look at clonal hematopoiesis, where you get mutation signature 5 in the blood stem cell, and you ask what is it that's changed a critical driver gene for that clone to get selection, and ask what signature is driving it, it's 80 to 90% of that is mutation signature 5.
So
The point being,
C to T and A to G.
it's C to T in the broad spread. It can be deconvoluted to that. And it has a very profound significance because it may suggest that the major driver for cancer predisposition or clonal expansion in us is signature 5. And all the things we think are mutagens from the outside are actually acting as promoters rather than actually driving the mutations. And there's a beautiful paper in bioarchives from the Sanger group that really does this at scale in 10,000 individuals, but from buccal smears, that essentially corroborates that. It's on bioarchives. If you're interested, I can send it to you.